Well, good morning, everyone. My name is Greg McMahon, and I am a principal scientist working at the National Physical Laboratory in Teddington, UK. And I'm going to be talking to you today about advances in using JUMP and JUMP Pro for analysis of high spatial resolution mass spectrometry images. So this talk is kind of a carryover of what I presented about 4 years ago in that ill-fated Munich meeting where COVID got in the way and turned it very quickly into an online meeting, and the ability of the folks at Jump to pivot and make that an online meeting a very successful event is still incredible. And so I'm glad to be with you all here today in person and online, and look forward to presenting my talk and talking with you throughout the week. So to begin, I'm going to be speaking about nanosims as our means of obtaining high spatial resolution mass spectrometry imaging. And there's a image of the instrument here on the left-hand side. There's only about 55 of them or so worldwide. They're used for various applications. Ours is probably about 80% biological, and then maybe 20% physical sciences, metallurgy, material science, semiconductor, if you will. But in a nutshell, very briefly, how does the instrument work? We generate a beam of ions, and we accelerate them and focus them towards our sample of interest. And when this beam of ions strikes the sample, the energy that is imparted into the sample causes atoms and molecules from the sample to be ejected from the sample surface. And in that process, a small fraction of those ejected species are actually ionized. And once they're ionized and carry a charge through application of high electrostatic fields, we can collect them and focus those down what we call a secondary column, and then into a magnet. So we use a magnetic sector mass spectrometer. And what it does is it separates those ions according to their mass to charge ratio in a manner very similar to how a prism separates light into its various wavelengths. And so once that separation occurs, we have what we call a multi-collection chamber, which houses 7 detectors. And these detectors, 6 of them are on motorized trolleys, so we can move them to any position that we like. And then the 7th detector is fixed at the sort of highest radius in the magnet. So typically our highest mass would go on that fixed detector, and then the other 6 are positioned accordingly. And in front of each detector, we can acquire what we call a high mass resolution mass spectrum, which we see here. It typically contains multiple peaks that are indicative of what we call mass interferences. I'm going to be talking a bit more about this towards the end of my talk. But really, the main advantages of this method are noted down at the bottom of the slide here. We get exceedingly high spatial resolution. We can push it down to about 35 nanometers. 100 nanometers is pretty typical. Doesn't require a whole lot of effort to achieve. We also get high mass resolution, M over delta M of about 15 to 20,000. So this enables us to separate peaks such as the ones that you see in the mass spectra here. And of course, because it's a mass spec technique or a SIMS technique, we call it secondary ion mass spectrometry. We get very high sensitivity down to ppm and ppb levels. So I'm going to talk to you today about a couple of case studies of how we've pushed forward. Last time I talked about how we apply it to cancer research. I won't be talking about that today. My first one is going to be a pretty simple example of how we have implemented the workflow builder into our workflow. And this has been really a godsend almost. So the first case study I'm going to talk about is it's about basically nitrogen fertilizers that we use to assist plant growth and their effect on climate change. So around the world, it really is the availability of fixed nitrogen that limits agricultural production. And unfortunately, a lot of times use is being made of these synthetic nitrogen fertilizers that can cause atmospheric pollution by ammonia and nitrous oxide in their production. They can also give off smog, fine particulate pollution also in the production. Once they're applied, they can actually cause ecosystem acidification and eutrophication of the waterways, and just generally are bad for climate change. And so what was discovered not too long ago, maybe 30, 40 years ago, was that there certain bacteria have this amazing ability to take inert nitrogen from the atmosphere, that N2, and they can actually fix it and convert it into ammonia, which can then be released into the plant for amino acid chlorophyll and protein synthesis. So effectively, it can basically replace those nasty synthetic nitrogen fertilizers we were talking about before. However, no one has ever actually shown this on a very sort of micro to nano scale in plants. There's been all kinds of bulk analyses done that show it, but we wanted to have a look at this in a lot more detail and at much higher spatial resolution. And so this particular demonstration is going to show how we have taken the leaves from a plant that has been grown, in this case corn, that has been grown in an atmosphere of 15 nitrogen, and no other synthetic fertilizer was given. There was no other source of 15N2 that could be possible. 15N, I should maybe backtrack a bit. 15N is a heavy isotope of nitrogen. The natural ratio is about 0.0037%. So what we're doing is we're growing it in almost 100% 15N2 instead of 0.0037N2. And then our idea is we're going to use the mass spec to hunt for 15N in the plant. And so I think at this point, I'll sort of summarize what our first level of analysis is and how we've used JMP Workflow Builder to get there. So our raw data files are actually quite simple. So that comes off as a CSV file that has 2 rows, X and Y, that define pixel locations, and then, 2 columns that define pixel locations, and then 7 columns or more or less that contain the counts from the various masses that we measure. And so typically the first thing that we would do is if we're looking at isotope ratios, we take the 2 masses that construct the ratio and insert a new column for the isotope ratio value. And then what we can do is we can go into Graph Builder and basically reconstruct the ion images, which we see here. So this image over here, this is the carbon plus nitrogen image of a section of the plant, and you see these various organelles. This here is a plant nucleus. These here are what we call chloroplasts. And this image over here is the nitrogen isotope ratio image, where we've got a color scale. So the blue, actually, the darkest blue represents the natural ratio, what we would expect regular nitrogen to show. And everything above that is ratios elevated in 15N that in this case could only possibly come from the 15N2 gas in which these plants were raised. So clearly we now have evidence that the bacteria, the seeds were inoculated with these bacteria, that they have done their job. They have fixed the N2 and distributed it throughout the plant. And we can also, of course, generate, it's very useful to look at the distributions of the various counts, especially looking at where the outliers are. And so I timed myself using how I used to do it, which is basically doing all these steps manually. And it took about 3 minutes to get to where I've shown here on the screen. But now with Workflow Builder, it takes roughly about 5 seconds. And, if we're looking at 20 of these odd files per day, that can be a serious savings in time, up to about 45 minutes to an hour per day, and over the course of a month and a year, that all adds up. So I'll just run a quick demo here um, to show you what this looks like. So if we, I'll just go open a recent file, and I'll pick this one. And so, as I said, this is what the raw file looks like. 2 columns, X, Y. These are the pixel locations and the counts in each of our masses. And now I'll go File, Open Recent, and Part 1 Workflow Builder. And there's our workflow builder. I'll maybe just shrink this a little bit. Okay. And then, let's just run it step by step. So a new column is inserted. It's given a title, nitrogen isotope ratio. A formula is given. That is, the ratio is calculated. Then we display our, that is our raw CN image. So that was the first image I showed you, except there it's colored on the slide. It was actually grayscale. There's the nitrogen isotope ratio image. Then we can finish off with the distributions, and then just the simple multivariate statistics with the scatter plot matrix and the heat map. And so what was, so we saw that the ratios were higher in these particular organelles here. These are the what we call the chloroplasts. Now, the chloroplasts are an organelle that is responsible for photosynthesis. And so that the nitrogen seems to be going there preferentially is almost a double win, because now we have the nitrogen preferentially going to support chloroplast growth and function. But the chloroplasts, those are the organelles that will take CO2 from the atmosphere and use it to convert it into energy that drives plant growth. So from a climate change perspective, this is actually a win-win. So we're lessening the need for synthetic nitrogen on our soils, and we're helping maybe taking up CO2 from the atmosphere. But while I'm here, I'm just going to do one other thing. So what was interesting was these high outliers in the sulfur signal. So we're just going to select them. And as you see, the selected ones have turned pink. These are mainly only occurring in the chloroplasts, some more than others. The reason for that, not quite sure. This might be a temporal effect. But this is significant because in order for that nitrogen fixation reaction to occur, it needs the assistance of an enzyme that happens to be rich in sulfur. So this was kind of an added bonus to the analysis of this particular sample. So that's just kind of a quick, first level analysis using Workflow Builder, and it just saves an immense deal of time. And so what I'm going to do next is I'm going to close out everything here, including that. Okay, and then head back to the PowerPoint. And then go on to the next slide. Yes, so this is the part where which I just showed you, with the sulfur preferentially into the chloroplasts, assisting that nitrogen fixation reaction to occur. Okay, so that was actually a pretty straightforward analysis, but sometimes we get others that require a lot more complexity. And so the second example I'm going to use is a study that we've carried out looking at what are called selenoproteins. And if you go to a health store, you've probably seen, a certain section where they have these selenium supplements. And it's kind of a very interesting field, in that it's still, I think, in its infancy. A lot is known, but a lot is still unknown about how selenoproteins work in the body. There have been, certain claims that have been made and evidence to support this, that they can be protective in the brain, be effective to help your immunity system. They may have antioxidant properties, help reproduction, assist your thyroid gland in its function, and also have these anti-mutagenic effects, which can help prevent damage occurring to your DNA. And so the one question we specifically wanted to address in this study was, do selenoproteins really associate with DNA to help protect the integrity of the DNA? And, recalling that we're looking at very high spatial resolution mass spectrometry imaging, that this is actually one of the few techniques that can do this and be reasonably effective at it. Now, the issue with selenoproteins is that they're already in very, very low abundance in your body. And, to make it easier, people might say, well, we'll just add a little bit more. But you really don't want to do that, because if you can also get selenium poisoning if you have too much in your body. So studying, samples in a petri dish that have had artificially too much selenium added is not terribly insightful. So what we did is we had some of these selenium precursors, and these would be typical ingredients that you would find on those bottles of supplements. We had culture media with the addition of 300 nanomolar of these isotope labeled precursors for selenoproteins, 76 selenium enriched methylselenocysteine, 77 selenium-enriched selenomethionine, or 82 sodium selenite. Now, I have to stress that everything, when I talk about isotopes here, these are all stable isotopes. These aren't radioactive or anything like that, so these are all perfectly safe. So we added these to the culture media, but we also added 15N thymidine. So this is our friend 15N, the heavy isotope of nitrogen. And this amino acid will selectively label the DNA. So what we're hoping to see here in this double label experiment is that we're going to see a correlation between where there's high nitrogen ratios and high selenium ratios, or vice versa, or maybe no correlation at all. So if we look at the initial images just coming off, so this would have been, if we used our work builder from before, what we find is that there are very low selenium counts, and this is probably not too unexpected, because remember, it is in low in abundance, and even by counting for as long as we did. So this image acquisition, this is the selenium ratio image on the left-hand side there. This was probably a day and a half to 2 days of image acquisition, just collecting counts from the sample as it's being rastered by the primary ion beam. And so when we do the ratio image, we see in the gray areas, we have all this missing data, and all these gray areas here, and there's probably tons in here that we can't see quite as easily. So we have basically two cells in this image. This is the carbon plus nitrogen image, so it basically looks at the sort of the biomass of the cells. Everything else, all this dark matter in between, is the embedding resin. So these samples are embedded in a polymer resin and then sliced at very thin thicknesses with a diamond knife. And we see we've got low counts. And so even before we go any further, we thought, well, we might be better off trading spatial resolution for more counts by binning the image. And I know now I'm starting to, jump into an image analysis software, which it was never meant to be. But it's having all this dynamic linking, just, the marriage between the two is superb. So I'm going to actually zip back and close this back to, because this, I have to thank Julian Paris for this. This was last year. We were discussing during lunchtime about how to bin images, and so I'm just going to go over that briefly. We've incorporated this into the workflow builder now, but so let me grab this image here. So this is the raw data, and the trick to binning it, it's kind of a two-step process. So I won't derive the isotope ratios here, but what we're going to do is we're going to select the X and Y columns, and then we're going to go down to utilities, and then we're going to make a binning formula. And then I'm going to go to cut points, and I'm going to bin this into 4x4 equal bin widths. So I'm going to go equal bins, offset 0 is fine, switch that to 4, I'll go OK, and make all like X. And then make formula columns. Okay. So there we have 2 more columns, X binned and Y binned. Then we go to tables. We go to a summary table, and I want to bin all the counts columns as well. So I'm going to select all those. And under statistics, I want to sum everything in there. So now I'm going to sum all the counts in the individual pixels into that one 4x4 bin. And then this was the part that wasn't quite so obvious, is we're going to group by X and Y. Then we go OK. And now we have our binned image. And we can just show that on the graph builder. Graph. And X, Y. And then we'll color, let's just color for now with our CN, because that'll be, and because it's binned, let's just make our marker size a little bit bigger, triple XL, there. And so now we have far more counts. It's a little busy with the axis scales here. But anyhow, I thought that was worthy of showing people in case anybody else is doing this kind of thing with their images, as it wasn't really quite that obvious when we started to work with it. So let me just close it out of here. And then back to PowerPoint. And, oops, current slide. Okay, so then we did, once we had the image binned, what we did was we derived the nitrogen isotope ratio and the selenium isotope ratio, and we got all our distributions and our heat map. And when we were looking at the heat map, something seemed just a little bit fishy. And that was that the selenium isotope ratio seemed to have a negative correlation with both the selenium, 77 selenium, and the 80 selenium. So we've got a negative correlation with both the numerator and the denominator, which just didn't seem right. You'd think it would be one and then the other, but no. So we started wondering, so what we were really curious about was all of these outliers on the selenium isotope ratio and the distribution. We also see a lot on the nitrogen isotope ratio too, but this doesn't bother us as much because we know the count rate is higher, and basically just through experience, we know we can get that kind of level of isotope ratio through 15N thymidine administration, and especially at the micromolar level. But this we weren't sure about. This is kind of uncharted territory. So what we then did was that we decided, okay, let's have a look at these outliers a little bit more a little bit more closely. So we did a robust fit outliers examination, and we found out there were actually, most of these were actually identified as being outliers through the robust fit algorithm. And so once we removed those and we redid the heat map and the scatter plot matrix, now things seem to make a little bit more sense. The 77 selenium had a very small positive correlation, and the 80 a slightly -1 for the ratio value. So that kind of made more sense. So at that point, we thought, okay, let's try doing a simple cluster analysis now. And so we did that. We did a simple k-means, just looking at three variables, very simple. So phosphorus, the selenium isotope ratio, and the nitrogen isotope ratio. And of course, what we were hoping to find, or at least according to our hypothesis, what there should be a cluster where we have both high selenium and high nitrogen in the instance where the selenoproteins are indeed associating with the DNA to help protect it against some sort of DNA damage. But we look at the parallel plots and we never see that. It's always one is higher than the other or vice versa, or they're roughly the same but kind of at a low level. And so this was kind of our first indication that maybe it's not working, or our hypothesis is incorrect. So when we looked at these outliers on the selenium isotope ratio, and of course we found that they were all associated with extremely low counts of 80 selenium and actually it should be down here, 77 selenium. And in fact, they were all embedded basically in the matrix or the embedding resin. And so this led us to think, okay, we have to take this another step further, because all this embedding resin is kind of biasing our analysis. So we got to get rid of it. And so, but that's easily done. We can just do a quick, another quick cluster analysis, basically just using CN pretty much and a couple others, and just do 2 clusters, and it'll be obvious through the high CN counts that we can segment all this out, and then just leaving the cells behind, and then we can do a table subset from that and have ourselves a new data table to work with. And that is what gives us this scatter plot matrix and heat map, where now we look at the selenium isotope ratio, and we see at 80, it's a negative correlation, or at 77, it's much stronger positive correlation. So now that's all making sense. And then we go, but the thing we're really interested in is the selenium isotope ratio and the nitrogen isotope ratio, which now shows a complete negative correlation. And so that means that, what we thought was happening wasn't. And so to look at, other areas, we would look at, hundreds of these cells. So we've now got our full workflow builder, which incorporates 4x4 bit of the image, using 3 times a graph builder for 3 different masses: the CN, the selenium isotope ratio, the nitrogen isotope ratio, the distributions, the multivariate statistics the first time around, an outlier analysis, the k-means clustering and the parallel coordinate plots, and then k-means again to segment out the cells. Then the new data table is created, graph builder again, then we can go on with multivariate statistics. And then at that point, we kind of finish there, but we've added another k-means clustering and parallel plot as well at that point. Now, it's might as well go show you how this works, because it is quite fun. So we'll go File, open Recent. And this is this file. And now we're going to go open Recent with the workflow builder. And that's part 2. That's that one. And we'll just launch it and watch it do its thing in real time. So here come the first set of images, second set, third, distributions, scatterplot matrix, outlier analysis. Next scatterplot, k-means clustering, data subset image, and finally, yeah, the last scatterplot matrix. So we can run that now with just right on the opening file. That saves an immense amount of time as well. So this is nice because I can, develop these things, send it to my colleague, and then when she looks at the data, she doesn't have to do all this. She just presses go, and it's all there for her. So let's close up everything. And okay, close that. Close. Nope. And so one last thing, so we're in January today, but by March, actually, this is the second last thing. Yes, I forgot. So after we finished all that, then we did more k-means clustering with that subset of data. And again, what we see with the parallel plots is there's absolutely no sign of high selenium and high nitrogen when we look at all the different clusters. And these are sort of, I would do minimum three, maximum 10 clusters, and then just use the one that was identified by the CCC criterion as being the best. But in every case, there's no sign of a high selenium and high nitrogen. And so what our conclusion is that at least directly, there is no evidence that the selenoproteins protect the DNA just by an association in spatial terms, if you will. But there still could be some indirect mechanism that they do so, but that's sort of well beyond the scope of what we were trying to do here. So lastly, as I said, so we're just the end of January here. Hopefully by March, I'll have a lot more to say about this, but this is how I think we can probably use Functional Data Explorer to help us with another type of analysis that we need. And that is I showed you at the very beginning, these high mass resolution spectra that are acquired at each detector. And we acquire these because when we set the detector, we need to know which mass we're actually analyzing because we have, in many cases, a lot of mass interferences. So what happens is, so I've just sort of blown up this region here at the detector end. So the separated ion beam passes through a set of deflection plates that scan the focused secondary ion beam through a narrow slit, and then into the detector. So the diameter of this beam is narrower than the width of that slit. So you can kind of see what happens when the deflection is quite large. It's, not really hitting the detector. It's hitting more of the edge of the slit and not getting to the detector. Then the deflecting path deviates it more, and it starts entering into the slit and on its way to the detector. So the counts start to rise. And then at a certain point, you're fully within the slit, and then you basically have a nice flat top peak, and then it starts going down, and then there might be a second peak from another mass that you might start seeing, and so that's what we have here, and then it goes down again, and finally the third. And so during an analysis, what we would do is we would pick which peak we would want to be on and set the deflection voltage such that only that peak or that peak gets in through the detector. And if this isn't set right, this means that all the data you acquire is basically useless. Now, for a lot of these, like for the 15N and we use 13C a lot, we know what these spectra look like. But for the selenium, that was really quite a challenge to sort it out to make sure we were actually on selenium, especially since the count rate was so low. But when I looked at this and I started looking at Functional Data Explorer, the first thing I saw when we were discussing the use of wavelets, is that these spectra look almost exactly like Haar wavelets. And another thing is that there's basically no background because we set our thresholds and our gains of the detectors to make sure that there is essentially no electronic background and that the response is the same. And we have to do this because, you know, we were measuring isotope ratios all the time, but we're measuring them using two different detectors. So if those two detectors aren't calibrated against each other precisely, our ability to measure a proper isotope ratio becomes diminished. And so basically all we need to know is the position of one of these and what it is assigned to. So in this case, I know this peak here, this broad peak is 12C and 14N. This one is part of the 12C, 13C, 1H, so these are both adding up to 26. And if we know what one is, we can sort of figure out what the other is just by difference in the masses. So this scale is actually, it doesn't matter because we're always looking at the mass differences. We could calibrate this to be precise all the time, but that takes an awful lot of effort and an awful lot of work. So if we know what one is, we can calculate what these other peaks are, differences, and then just associate back to a program that we have. Essentially, we can put in all the elements that we think is in the sample, and it will fire out all the possible outcomes that add up to that mass, and then the associated difference from that particular peak. So if we could use FDE to sort of separate all these out, kind of like a deconvolution, this would be a massive boon for us, particularly in some of these heavier masses where we get more interferences and they're much more difficult to sort out. And so that's pretty much all I have to say about this at this point. But by the time the meeting rolls around, I'll have hopefully a lot more and maybe some examples as well. But for now, I would just like to acknowledge some colleagues, Dr. David Dent, who's at the Sustainable Nitrogen Foundation, and Professor Eric Murchie and Professor Ted Cocking at the University of Nottingham. They were important in the first case study on the nitrogen fixation by the GD bacteria. Dr. Diane Handy and Professor Claude Lechen at Harvard Medical School. They were, essential for the selenoprotein study as well, and of course all my colleagues at NICE MSI at NPL. And on a sad note, I'd like to just say that Ted and Claude have since passed away. It's a huge loss for the scientific community, but they're remembered on with this talk and many others to come. So thank you very much. And if you have any questions, I'd be pleased to answer them for you.